Dear audience, I'm going to present the topic of my PhD studies, which is investigating the role of probiotics in shaping the human gut microbiome. My name is Éliás, and I'm a dietitian for profession working currently at the Faculty of Health Sciences at Semmelweis University. My aim is to formulate evidence-based recommendations for the use of probiotics in several conditions, and for that, my aim is to understand the role of the human gut microbiome in health and disease and the consequences of treatments on that. In the framework of my PhD studies, I will investigate the role of probiotics in several conditions, first during antibiotic therapy, and after that, the supplementation of probiotics in the healthy population. So the topic of my first project is investigating the efficacy of probiotic administration for the antibiotics-associated dysbiosis. Probiotic dietary supplements represent a $1 billion market, which is still expected to be growing. Probiotics are used in several conditions. However, we know that these products are 9 times more likely to be used during antibiotic treatment. We know that antibiotics has a major impact on the human gut microbiome, and it is one of the main factors leading to the so-called low diversity dysbiosis, which is a potentially harmful state, and it has been associated with acute diarrheal disease, but even with Clostridium difficile infection and inflammatory bowel diseases. Probiotics are widely used to prevent this dysbiotic state. However, the current knowledge has not been sufficiently summarized regarding the clear impact of probiotics on the post-antibiotic dysbiosis. Therefore, our aim was to summarize the currently available evidence using the currently available randomized control trials data. To formulate our clinical question, we used the PICO framework, so we investigated patients during antibiotic therapy, either supplemented with probiotics or not, and our main outcome was the gut microbial diversity and also the taxonomic composition. And according to our hypothesis, probiotic administration has a significant impact on the post-antibiotic composition and diversity of the gut microbiome. Here you can see the flowchart of the systematic search and selection. We screened nearly 12,000 articles, and in the end, we were able to include 15 articles in the systematic review and 4 of them in the meta-analysis. Here you can see the results of the meta-analysis of Shannon Diversity Index. And as you can see, first we analyzed 4 articles where we showed the significant effect of probiotics during antibiotic therapy with a mean difference of 0.33. But we also performed a subgroup analysis, because you can see in the first forest plot that we had an outlier article, and the reason behind it that this article investigated a basically healthy population without any medical reason for the antibiotic treatment. Therefore, we performed this subgroup analysis with patients with medical indication for the therapy. And here you can see that we had an even more pronounced effect of probiotics in maintaining the diversity of gut microbiome during the antibiotic therapy. Sadly, we were not able to analyze more diversity indices, which can be used for describing the gut microbiome diversity, but you can see that we have several of that. Sadly, the current literature does not have a gold standard for that, for which is the best index, so-called best index to use. So it is really heterogeneous in the literature right now. But here you can see that we were even not able to include all the articles that were reporting on the Shannon Diversity Index in the meta-analysis due to qualitative data reporting. And in other cases, we didn't have enough article to perform the meta-analysis, but I would highlight that here we have more controversial data or results regarding maintaining diversity, because the black cells, you can see it represent the cases where actually researchers didn't find any significant difference between the groups. So in conclusion, we can see that our meta-analysis suggests that probiotics can have a significant effect on the gut microbial diversity during antibiotic therapy. However, the qualitative data synthesis has controversial results. The strength of our study is that it is the first meta-analysis performed in the topic, and it represents the highest level of evidence since we only included randomized control trials and we used high-quality methods. However, we have some limitations. First of all, the low number of studies and also the low sample sizes, and the qualitative data reporting, and also the heterogeneity of the studies. Therefore, we could not perform more meta-analysis in the topic, and also the studies were heterogeneous regarding the reason or the indication for the antibiotic therapy, and also the duration, the type of treatment, and so on. So the implication for practice could be that we can say that probiotics may be beneficial for maintaining the diversity of gut microbiome during the antibiotic treatment, but I think we have the highest implication regarding research, because we see that we really need more randomized control trials with standard units of measurement so that the data can be quantitatively analyzed together. And we also need clear cutoff values for the ideal values of these diversity indices, and also the evaluation of correlation between the clinical manifestations and microbial composition and diversity indices would be important. We are soon finishing the manuscript, and I had the chance to present my data in several occasions and some conferences. So we are aiming to publish our data soon in the journal Gut Microbes. The second project of mine is investigating the effects of probiotic supplementation on the gut microbiome diversity and composition, and also on other clinical outcomes in the healthy population. As I mentioned earlier, probiotics dietary supplements is a really big market, which products are marketed even toward healthy individuals without any medical reasons, and their effect is not yet clarified for these healthy individuals. Therefore, we aimed to summarize the data currently available on that. So we will investigate the general healthy population, either supplemented with probiotics or not, and our main outcome is, again, the changes in the gut microbial composition, but we will also report any data, any clinical parameter in each study reported. And according to our hypothesis, probiotic supplementation will not alter the composition of the gut flora, but it may have an impact on other health outcomes. We finished the title and abstract selection, and we have a huge number of articles remained in our pool. So we are we will continue the full text selection. However, we can see now that because we included all clinical outcomes basically that were mentioned in the articles, we have really heterogeneous outcomes groups. And therefore, we will see how many articles we will have at the end of the full text selection, and maybe we will divide it even in more articles if we cannot summarize it in one, and we will contact probably other health areas professionals to help with these data. So I am going to submit the first article during September, and we aim to submit an article regarding the second project as well during this year. So thank you very much for your attention, and I am happy to answer any question that arise. Thank you.
Thank you, Anna, for the excellent presentation. I would like to ask regarding your first project. You mentioned probiotic supplementation. However, could you give us a little bit more detail on what it looked like in the 4 or 3 different studies that you included? Was it standardized, or did it differ by any chance?
Of course, thank you very much for your question. Of course, it was not standardized regarding the probiotic supplementation either. I mentioned that the antibiotic was not the same either, and also the probiotics differed. Yes, so which is the same basically, that it was an oral supplementation, so not in food, so not probiotic food was provided, so not yogurt, but using like tablets or capsules. So the form of the supplementation was that, but the strains, the colony forming units in it was sadly different, so yes.
What about the length of the supplementation?
Yes, I can show you. Here you can see the treatment period column, so it differed as well, of course, but actually it refers on the treatment period like it was the duration of the antibiotic treatment, and these patients were supplemented the whole duration of the treatment, so the concurrent treatment, so-called, was like this. So these days was the duration, but in some cases they also continued the supplementation with probiotics, but since we didn't have enough data to compare these results, therefore I could not show you any other details on that.
Do you have any data that tells us how should we be taking these probiotics in relation to the antibiotics at the same time, half an hour apart, 6 hours apart? Because one would imagine that antibiotic kills the probiotic as well. So is there any data on that?
Yes, actually, thank you for your question. It is, again, not standardized in the studies, but yes, they didn't take it at the same time with the antibiotics, but it differed how much was the delay, so like an hour later or a half day later, so it differed, but not at the same time.
And you couldn't see any obvious difference in the outcome in terms of the delay?
We didn't have enough data to concentrate on this question specifically, but probably not.